Good morning, everyone. Hi. I knew there'd be more for today's section now that a quiz is sort of looming on the horizon. That's on the 13th, right? So we're finishing essentially, our last, 10 days of this module. And for today, what I want to talk about are the Mendel's laws of independent assortment and segregation, which are the most important components of meiosis. That's really where a lot of variation is generated. But before I go into that, I want to see how everyone's doing. You have any questions or concerns before we go in today's discussion? Nope, everyone's good. All right, I like that. Okay. Additionally, what I'll do is I'll talk a little about the extra credit assignment to give some hints on that too, because I really do like to see everybody do those because, you can pick up 10 easy points that way and have some fun watching them. Pretty good visual stuff that I want you to do. There's nothing difficult about it, right? All right, let's do a screen share on, there you go. PowerPoints I have posted on there. All right. So from your work so far and what I've mentioned, remember when we're talking about meiosis, we're talking about the replication of sex cells, not somatic cells. I think that's a really important distinction because, on the quiz, I'm likely to put some sort of, definitive terms that you should know. And if you understand the difference between meiosis and mitosis, or at least where they take place at, you've probably gotten a point there. So I want you to be aware of that. So meiosis, talking about the division of sex cells. And this is the only place in which evolution happens, in which variation is actually established, because the products of these things are gametes, right? They're the individual gametes that go into reproduction. Okay? So what I like about this too, learning about it, is that if you learn meiosis, it doesn't matter if you're looking at humans and turtles and bears or squirrels, or even a creature such as Justin Bieber, which are very bizarre. You're learning about the same thing. The process doesn't change across species. Everything's linked together because everything is life, right? Okay. So I mentioned this before too. It's important to look at these things as a karyotype. In other words, these are all of your chromosomes, which are on display. And if you look at some of the labs already, realize that there's different numbers of chromosomes in different species, and it depends on what their variation needs are, and it also depends a lot bit on luck too. When chromosomes are actually replicating or they're dividing, we could have all sorts of problems in their divisions to where we can over-replicate, we can truncate these things. And if it actually works out, they can go into, sexual reproduction and be permanent part of a species. So any change that happen in chromosome and chromosome numbers across time are, of course, results in mutations. And then they're, sort of up for trial and error to see if they're beneficial to a particular species or not. Ours is set at 23 pairs, which seems to be fairly optimum for our species. It generates an adequate amount of generation for us. And it also reduces a number of mistakes. Remember, the more chromosomes that you have, the more those things need to be divided and packaged into gametes, and the more actually mechanical errors which are actually possible. Thus, by keeping the number of chromosomes down, we sort of risk, mechanical errors. And for, a species such as humans, those are very important because reproduction is so expensive for us. Not only gestation time is 9 months, the amount of energy that it takes, to raise a kid just through gestation, the risk to females and the rest of the society, and as well as how long it takes to raise a human being. We actually don't have as much variation as many other species, and we've thought about that for a while. Why is it that humans show a remarkable lack of variability? Of course, we have more variability than a shark. They're almost the most invariable sort of things we see, these chondrichthyes, these cartilaginous for fish. But for humans, it's because of the plasticity of our behavior. Let's remember that as we adapt to our environment, it's not so much physically, right? So if you're going to go to Mars, or if you're going to, like William Shatner is going to go to space next week, Star Trek guy, 90 years old, he's not going to grow, hair all over his body, What he's going to do, he's going to go to REI, he's going to get some high-tech equipment, and that will instantly adapt him to different conditions, right? So as a cultural species, we don't need to have the type of variation that other species are, because we're not, adapting to, climatic fluctuations, because our culture can actually do that for us. And in a way that gives us not only a behavioral advantage to be able to do that, It also allows us not to have as much variation and risks the outcome for each individual. I won't belabor this anymore. So a karyotype is also a snapshot of the chromosomes right when the cell begins to go into cellular division of meiosis, right when your sex cells begin to divide. So we're ending interphase, beginning to go into the other phases of meiosis, and the chromosomes are nicely packaged together, getting ready to be divided, right, to be copied, and then placed out in their individual gametes. The only time we actually see them like this. And we know the first 22 are known as they're known as autosomes, because they just really produce proteins that to go into growth and development and maintaining the body throughout the lifespan. And the number 23rd one, which we can see over there at the very bottom right-hand corner, are your sex chromosomes. And this one here, because it's got a Y, is a male. And if it was a female, it would have 2 X's. And I think I mentioned this before too, that being a male is not a big deal. Because males, whatever male is, it's culturally defined. I don't know, if you're male, you think you have this behavior that's male and you're so radically different from female. That's your culture that actually creates that through the enculturation process. And if you don't believe me, take off your shirt and look in front of you and go, what the hell are these things sitting in front of me right now? Okay, right? You're a barely differentiated female. Right? And a lot of times females don't want to see that either, right? Because we want a strict dichotomy between sexes many times, depending on how you're raised. I'm like most people, I would say more hetero-cisgendered, right? But that doesn't mean that's the reality of our species, right? Genetically, we're a lot more complicated than that because there's so much overlap between what male and females is. And you can see it right here, because the majority of what I am is on that X chromosome, the female side of the chromosome. The X just gives it a slightly different signal. Okay. And that'll be important for us because we can look at what we call X-linked traits. Because the one thing that males always receive in their inheritance is an X chromosome. That's something that females don't receive. They don't receive the genes on the Y. So imagine this situation. So I'm an inheritor of an X chromosome. Okay? And the X chromosome contains more genes other than just determining sexual characteristic. That's why it's so large, right? There's other genes on there that go into growth and development. If I receive a recessive condition that's bad on the X chromosome, I don't have another X to balance that off. I don't have an X that can have a dominant healthy chromosome on it. Therefore, I will receive what's called an X-linked disease, genetic disease, right? Where a female, your chances are much smaller because you have 2 X's, and most likely the dominant one is the one in which will provide you the correct and healthy gene. Therefore, you'll slip many sorts of disease situations that males will have. And one of the reasons for that is males are expendable. That's just the way that it is, because we have a higher reproductive capacity than females do, right? We do. The members of a species with the lowest reproductive capacity are the most valuable ones. And here's an example of what I mean by that. Ladies, so when you think about your sexual lives, which, whatever it is, think about the duration of it, which you have, okay? So how many weeks a month are you receptive? Not the full month, right? Because of menstruation. What about males? All the time. What about when you're pregnant? But you're down, right? Not males, they keep going all the time, right? And what about after you're, say, 50 or so when they go through menopause? Well, males, we have the rest of 30 years to keep going. Therefore, we have a much higher reproductive capacity. Thus, we have to compete for females, which have the lowest reproductive capacity. Therefore, you are the actually most valuable members of the species, and you will see that cross-culturally in lifespan. Females always tend to live longer than males, right? And one of these situations is here genetically because you have another X, but you have other advantages too, which we will do a little bit later in class. Okay, so one of the first laws that Mendel determined, and we can see it here, and I kind of want to start printing Mendel's sort of influence, and I'll do a little number on him in a second. But Mendel said that, and he called his law of segregation. When he began to look at pea plants, when he began to experiment with these back in the 1880s, he noticed that when he was breeding plants together, that there were a couple of different things that could actually happen if he was looking at what we call monogenic traits. Monogenic traits means just one gene, one effect, right? One, you know, protein coding gene, one effect. And of course, Mendel didn't know anything about proteins at this point. He just thought they were traits. So what he was doing, he was like taking pea plants that were tall and pea plants that were short, you know, and the pea plants that were tall were all what we call true breeders. And that means that he would take these peas from a plot, you know, on the countryside, that generation after generation after generation only produced tall plants. So whatever their genetics were, they were limited to their own. They hadn't been contaminated or crossed over by anything else. And then he had, you know, short plants, which were true breeders. And of course, they were isolated and they only had the same sort of genes. Well, then, you know, he brought them together, you know, they pollinated each other from big distance. So he brought the plants closer together in the Abbey, the St. Thomas Abbey, which he was doing these experiments. And he was looking for results. What would happen? Oh, what he found was a system of dominance and recessivity, and a system where it takes two factors that he said, right, to create a genome, to create the traits in which you have. And he called this his law of segregation. So you think about two genes, they can be the same or different, and they have to come together to create an organism. That's when we're talking about laws, Mendel's law of segregation. Right? So the segregation laws say this, that the two genes that are coming together from the individual parents in sexual union can be the same genes. They can be the same genes. And by the same genes are the same alleles, because when they come together, because the potential that they could be different, we throw in the term allele. Okay? Allele doesn't say they have to be different. Allele says they could be different. So in some cases, they can be the same. The alleles are the same. Now, if they're dominant alleles, that means these alleles want to express themselves. Then we have a genetic condition known as homozygous dominant. Okay? Homozygous dominant. So whatever those alleles are coming together, they're going to express because they're dominant and the same thing. Now there's a different situation, which is also a homozygous situation where each of the parents will contribute, you know, a trait, but the trait's the same, but the trait is recessive. And recessive means that it generally will not express itself if it's just alone. So if it's on a chromosome alone, and there is a dominant chromosome adjacent to it that comes together in sexual union, which we call heterozygous, the recessive condition will not express itself, and only the dominant will, and there's your heterozygous condition. But to mention, if you have two recessives that come together, that is the only time in which the recessive will show itself. It will express itself, and that condition is homozygous recessive. Now that's part of Mendel's law of segregation. So when we say law of segregation, really you have to think about the components involved in which there's essentially three. Now, your traits are controlled by two unit factors. Now, these unit factors, what Mendel did was he was very smart about these. He goes, you know what? I'm just going to call them a ubiquitous unit factor because I can't see them, and he never did. There wasn't a light microscope at the time of Mendel. And Mendel said it wouldn't be scientifically responsible for me to say what they are until I can at least see them, right? And it wasn't until, you know, roughly around 1902 that people started seeing these things and labeled them actually as genes. I think that's very scientifically responsible itself. He said, it appears there's two things. Okay, that's what his pea plants were showing. There are two unit factors. And these unit factors must be together for the gene to express. So it takes two. There are two and it takes two. And these unit factors can be same or different. And one may be dominant and one may be recessive to the other. So I want you to think about that complement when we talk about his law of segregation. Okay. All right. So the little thing I have in front of you to decipher that, you know, little stubby things there. Just this thing of the little lines there is just one gene along, you know, a chromosome. So, you know, like say chromosome number 1 may have, I don't know, 2,000 different genes, protein coding genes on it. And the 23,000 genes that it takes to make a human, roughly 23,200 genes, are distributed out across the 23 chromosome. So about 2,000 on number one, because it is a larger chromosome. So there's 2,000 little bands there, right? And they come in pairs all the way down those pairs. And this is just like a tiny little slice of that. Right? Okay, so, we talk about, meiosis versus mitosis. We're not going to talk about the differences because that's interesting if you're in a biology class. It's one of those foundational things that you want to do. But I'll tell you what, in the world of anthropology, when you really sort of get into evolutionary history and stuff, mitosis, the division of body cells, it becomes completely uninteresting because nothing that happens, nothing that changes in the DNA during mitosis can ever be passed on. It can only affect the individual, yourself, the living organism. So the processes, while they appear differently, you can see the naming of these. You see the naming, like in mitosis, it's called interphase, prophase, metaphase, anaphase, and telophase. These sort of phases do actually exist in meiosis too. But if the problem is that that's not the way cells really go about their lives. There is no sign on the cell that says, I am now in interphase, red light blinking, okay, and I'll turn the white light on because now we're going to prophase. There is no definite sort of move from one phase to another. It's all a smooth sort of continuous thing that happens when we're talking about cellular division, mitosis or meiosis. Humans have a need to put categories on things. And we think in terms of container, like your life, half full, half empty, right? And think of the analogies that we use that are always about measure and assortment. And it just reflects the underlying, structure of our mind and the way we learn, which means we can actually have false conceptualizations about the world too. I'd rather us think about it, in terms of process. So let's look under meiosis. And let's look at the processes which are happening there. Once you hold these in your mind, you can see the flow of what's going on. So there are 6 basic processes in meiosis. One we call Tetrad Formation, crossing over, recombination, first reductive division, second mitotic division, and it's over, okay? So I want us to go through that and see, how this works and really where evolution's happening now. Where's the crux of it? And also, additionally, where is Mendel's most important law, okay? Segregation is his first law, but his most important law is his law of independent assortment. And that's the one I really want you guys to know. Okay? All right. So, more terms. You guys should have the terms down by now. But just to let you know that, when we're talking about chromosomes, we have 23 pairs, right? When you have full pairs, together, okay, full pairs, 23. That means there's 46 total. And each member of a chromosome pair are called chromatids, sister chromatids. Okay, so one chromatid comes from mom, one chromatid comes to dad, and you have a chromosome. So you have 46 of these things together. And when you have them together in 23 pairs, you have a living organism, and that's called a diploid condition. So when you see something in diploid condition, it's the living organism. It's the whole organism working together, eukaryotic cells and all. Now, if you only have half that, in other words, there's only one chromatid from a chromosome pair present. There's just 23 of them. Then you're looking at a gamete, okay? That's it. It's a gamete. It's not a living, it's not a living being. It's a reproductive cell, and it's in the haploid condition. So associate haploid with gametes only, and associate diploid with the full organism only. I think that's a good conceptual way to look at this. Gametes, again, are reproductive cells. And once those reproductive cells come together, they create a diploid organism. It's called a fertilized egg that begins to grow and develop into you. So a human life cycle is very simple. We go from haploid to diploid, to haploid, to diploid, haploid to diploid, right? So that has been our reproductive cycle since we've been anatomically modern for the last 100,000 years. And essentially, most likely, we think this system's been in place, for nearly 3.5 billion years, is a reproductive strategy that links us together with all other creatures on the planet. There are some variations to this. There's some interesting, ways in which some plants and fungi do things a little bit differently. And you've probably seen these things with tadpoles, which go through some interesting transformations as they become frogs or metamorphosize with butterflies. and they have some intermittent sort of conditions, but they're very rare and they don't speak of animals. So we'll stay, you know, within our kingdom here. All right, so, you know, people say, well, Mercer, so why do we do this thing called, you know, meiosis? Why do you need to, you know, reduce the chromosomes from a, you know, a diploid condition? And that's what your cells start out with. When we talk about a primary sex cell, there's a certain number, there's a few primary sex cells in your gonads. For males, they are right now. Ladies at your age, they're all gone. What happens is all the primary sex cells in your gonads, in your ovaries, have already divided out by the time you're 13 months old. They're done. All the eggs that are in your body are there and done, and you're not going to get any more. They're all stored in your fallopian tubes each side, right? And they alternate until they run out, and you turn, you know, almost 50, and that's the end of it. Now with males, it's different. But those primary sex cells continually dividing or producing gametes, you know, all their lives. But it's only a matter of timing. But essentially, the reasons and the processes are exactly the same. So if the primary sex cell begins out as a diploid cell, first, the primary one, that's the one's right in the center, that's the nucleus cell, that's the one, right? Well, you know, if it copied itself completely, like mitosis does, that means it would produce another diploid cell, right? So if it produces a diploid cell and it sends that off as a gamete, and then it fertilizes a female cell who may be the same way, who is just another diploid cell. What do you have? You have a double diploid cell. So you've gone from 46 chromosomes, which are 46, you know, total, right? 23 pairs, to 92 in the next generation. Well, if the next generation does that, it doesn't reduce the number of chromosomes. If it's sex cells, which are, you know, now 92 chromosomes each, right? Go into reproduction together. Look at how many chromosomes the next generation has, and the next generation, the next generation. By now, through time, we would have billions of different pairs of chromosomes within our cells, and we would literally explode because there's way too much genetic information. And in fact, we wouldn't even operate. Anytime you exceed the number of chromosomes you're supposed to have, you're going to end up with severe genetic abnormalities. For example, there is a genetic disorder called trisomy 21. When the cells end up with an extra chromosome number 21, then it does cause some mental impairment, some damaged developmental issues. It's called Down syndrome. So it's quite serious when we don't get the right amount of chromosomes that we're supposed to have, right? So that's one of the reasons that we make sure that when that primary sex cell does begin to replicate, it reduces its DNA in half. So it can only, you know, contribute 23 chromosomes. Now, the next reason it's doing this, of course, is more important for us, meiosis, is that when it's reducing its chromosomes, it also needs to replicate its chromosomes. So we're talking about a process of replication and reduction. And it's in the replication process of the DNA that all evolution happens. We have to remember that too. Evolution doesn't happen anywhere except the very first part of meiosis, when that primary sex cell replicates its DNA. After that, the process of reducing the DNA, right, trying to get it down just to 23 single chromatids, is a product of randomizing all those mistakes that come from the DNA replication process itself, and randomizing it in all sorts of different combinations to go in all sorts of different sorts of gametes to, you know, come together in sexual union to create wholly new forms of life which have never been seen before and absolutely unique, for the chance that some of those forms of life may have some unique characteristics that will allow them enhanced fitness in the environment in which they find themselves when they're born. Okay, so one thing I mentioned to you before, I gotta hit it again. It's so important. Your lab book's gonna go into detail about this, but I'll tell you what you really need to know when talking about the first part of meiosis, when all evolution happens. That the division of DNA, replication of DNA, is an extraordinarily simple process. As I mentioned before, all processes in nature are simple, right? We take the initial strand of DNA, you know, and remember there's going to be, you know, 2 strands of DNA in each cell, right? So number 1 chromosome, there's mom's strand of DNA, chromatid, and there's dad's strand of DNA chromatid in each one of your cells, right? And for chromosome number 2, the same thing. Number 3, number 4, all the way to 23. Pairs of these things are doing this. So each chromatid is unwinding, separating, right? Because it's a double-stranded molecule. They're separating. Then each of those strands acts as a template. In other words, one strand remains there, and the other complement is just copied, built, right, on the founding strand, or it's where the parental strand, right, the template strand on each side. So you get two, strands of DNA when before there was just one. So this little diagram shows you essentially what's happening. The copying process is a little bit more complex than that. There's a couple mechanical things that if you were in a biochemistry class that we talk about, and in a lab manual, Catherine Slurry does speak about it a little bit, about copying can only go in one direction. So as you're unwind the DNA, one copying process goes up while the other one's going down. So you have to keep adding little fragments on one side as it's unwinding. But those are just small mechanical issues. We don't want to get buried in that. What we want to think about is this. When those strands are unwinding and another strand is being added, there's pairing rules. We can actually predict what that strand is going to look like. Because we know in DNA, remember, A always pairs with T, C always pairs with G. So as those strands are unwound, we can see the base pairs which are exposed, which are A, T, C, or G on one strand. And we already know what the body is going to do. It's going to build the complement strand all the way up. Here's the thing. What if the body doesn't do that? What if the body makes a mistake? Instead of pairing an A with a T, it actually changes the A and puts a C in and pairs a C with a G. And we can see that here in this little chart over here to the left, where it says normal DNA on the top. Okay, so imagine the very top layer is like a sequence of DNA. You're reading, down the DNA, TAT, CAT, CCT, AAG, CTA. And the body is now going to try to provide a complement. It unzips this strand, puts the complement to it. And when it does, it changes one of the letters. It makes a substitution. And in this one, we can see the red letter there that is substituted. It changed the C to a G. And when it did that, it changed one amino acid. So that protein, that gene is now coding for a sequence of amino acids to produce a protein. That's no longer tyrosine, histidine, proline, lysine, valine, but it's now tyrosine, histidine, arginine, lysine, and valine. That is called evolution. That's it. If you change that code, if you change the amino acid sequence, you're going to change structure and the function of the protein. Now, there are different sorts of patterns that we see. If I change just one letter, substitute one letter, we call those point mutations. Point mutations. It's just one point that's changed. Now, I can do all sorts of point mutations, okay? This one's called a substitution, but I can also do an insertion too. So in other words, what happens if I decided maybe not to change a letter, but to insert a letter? What would happen is you have two particular options. If I insert a letter into the genome, just one letter, think about it, okay? So if every 3 letters represents one amino acid, okay, and you're going in sequence 3, 3, 3, 3, 3, 3, 3, 3, and then I randomly just insert a letter somewhere, think about what could happen. Think about changing the reading frame. So it will shift over one letter, right? So if I add a letter, it throws the reading frame also. I'm not just doing the same 3 amino acids, I'm actually shifting one letter. So that means every single amino acid is now going to be different downline from where that insertion was at. So if I do that and I change the reading frame, I can have a dramatic shift in the protein, because almost every single amino acid from where I inserted one letter, it will be different all the way down. That's a radical step. Now, compare that to a simple substitution where I just change one letter, not add a letter, but just change one letter. If I change one letter, I'm just changing one amino acid. If I insert a letter, I'm changing all the amino acids down. That's an insertion. How about this? What if I inserted two letters? What do you guys think would happen? Inserted two letters. Same thing. I would change the reading frame all the way down line. Everything would be different. From wherever that insertion of two letters were, it would change the reading frame. The three that are being read would be completely different. A huge change in the structure and function of the protein. How about this? What if I inserted three letters? All I would do is be inserting one amino acid. I wouldn't be changing the reading frame. I'd end up with the same protein with an addition of amino acid, okay? An addition of amino acid. Now, think about what's the purpose of something like this? To allow multiple different degrees of variability. Okay? So the body does simple substitutions, right? Just simple point changes, like just change one letter. Minor change will happen to the protein. Minor shifts in evolution, minor. Okay, now it can up the ante, right? It can do like, it can insert three whole letters. All of a sudden, I've inserted a whole new amino acid there, wasn't there before, but the rest of the amino acids are the same. Well, that's the next level of sort of impact, bigger change in the system. And what if I want to throw in some big risks to see what might happen? Well, I just throw in one letter at a time. I insert one letter, which changes the entire reading frame downline of that letter, or I insert two letters, which changes the reading frame. So now we've seen, a couple of patterns that can happen. Now there is another pattern, but it'll be sort of a familiar friend to us. We can delete. We can do the same thing in delete. So if I, delete a single amino acid, one letter. I'm sorry, not one amino acid, one letter. If I delete just one letter, I change the reading frame. So everything up to the point of deletion is normal, but after that, I change the reading frame because I got to go three letters at a time. If I take one out, it just changes the three that I'm looking at. So all the amino acids are going to be different downline of that. Well, same thing. If I just take away two different letters, I change the reading frame. So every three are going to be different. It's going to shift over. So whatever's downline of where that change was, all the rest of them are going to be different amino acids. Huge change, right? So between these single point mutations, which may just substitute, one single letter, maybe changing one amino acid, from that to the deletion of letters, one or two, which are going to cause major frame shifts. Body can control the types, the severity of mutations in which are going to happen. And sometimes I think, the greater the risk, the greater reward. I think your greatest pleasure are on the other side of danger. I don't know if you guys know that, but that's part of you. Sometimes you got to risk, things, your greatest pleasures are. Well, that's the way evolution is too. That for an organism, it can be a big risk because when you're talking about frame shifts, they can really cause disabilities and problems, even miscarriages. And in fact, at least I don't know if you know this, but 30% of all your pregnancies will become miscarriages because of this process. These sorts of mutations are going to happen. And all of a sudden, you're going to, through mutation, you're going to change your gene radically, that it won't even work anymore. And when it's necessary for the gestating fetus, whatever phase it goes through, and that protein's not there, arrested development in the womb, the genetic system senses it, and bam, it cuts it off the proteins, cuts it out of the placenta, and you get a discharge, and you're going to get a miscarriage. Miscarriages are normal, by the way, 30%, because of this development process, right? Because body throws in big sort of changes every once in a while. Now we can question, this, why big changes? Well, of course, you need to have big changes because sometimes, life's like that. Other species are making big changes, and sometimes they're going to give the jump, the big advantage on other species. And while it may be a risk, to the fetus and the downline itself, evolution is not about individuals. It has nothing to do with you and me. Your life means something to you. To evolution, it doesn't. Evolution is concerned about the health of the species and not of the individuals. Okay? All right, so now you can see how DNA divides itself, some of the patterns of mistakes that we see. These are about the only types that we see. Now, however, there are things that can actually happen that aren't associated with our own copying processes. These are things that happen right now in the viral world. One of the papers I want you guys to read are, paleoviruses and human evolution, because when viruses invade you, they do put their own DNA and RNA in your genome. That's how they do it. Their proteins are designed to insert their DNA and RNA into your genome. And then you go through transcription, translation, and produce the viral proteins that are assembled by your Golgi bodies, right? And they will destroy your cell. You're actually creating, your own doom, the things which will kill you, right? But, if these viral proteins are inserted and something goes wrong and the virus doesn't thrive, and that happens within your sex cells, those inserted pieces of DNA can be replicated and put on into your downline generation. And many times those sequences in which viruses embed in you are going to be producing proteins, right? They can be beneficial to you, or they can also be negative to you. They can be cancer causing. In fact, one of the leading causes of cancer we're finding out are embedded DNA and RNA sequences within our own genome, right? That carry oncogenes that cause our systems to actually become genetically go haywire. So all this bad press about the chemicals in our environment, which is true, sometimes we have to sort of look at the other forms of life, such as viruses, who are big culprits in this also. Okay, so let's move on. All right, so what happens during this process of meiosis? What are the important things that are going on? Well, first, that the DNA has to replicate itself. So here we have an image on the top where DNA has replicated itself. So you see, formerly there would have been just, one chromosome, and there'll be a red side and a blue side, okay, a red side and a blue side, right? And that's your normal genome. There'll be just a diploid. But what happens in the first part of meiosis is the chromosomes replicate themselves both sides. So now you have, two mom chromatids and two dad chromatids. And that's just chromosome 1. This is say a chromosome 2 or chromosome 3. That's just one chromosome we're looking at. So now you have twice the amount of DNA that you need in your cell when we go through meiosis, the first part, which is the interesting part. We go from a normal diploid cell to a double diploid cell. Well, why are we doing this? Why would the body do this? Well, to begin the process of acquiring variation. Remember when the DNA divided, right? So you see two reds. That's mom divided. She just divided. She created another red of herself, and dad just divided, creating a blue of himself. When that happened, they threw in 30 to 40 different mistakes that were never there before when they divided each one of the chromosomes. So now you got 60 different mistakes that were not there before in your genome. Now what you want to do is we want to randomize those mistakes. So the chromosomes come together, they tie themselves off. And because there's 4 chromatids, right? 2 moms or dads, we call it a tetrad. They come together, they tie themselves off in the center, right in the center, right? And then the arms of the chromosomes, proteins surround the arms and actually move the arms around each other. It's disgusting. It's like this for real sex happens. I can't look, I'm too squeamish. No. They do. It's like they wrap their arms around each other. And they touch each other. Oh my God, they touch each other, right? Cellular sex. Don't look if you're underage. Stop that. No, right? They touch each other, right? And when they do, they're exactly in the same points where the same alleles are. So there's an allele for blue eyes, an allele for brown eyes right there. They touch themselves right where those alleles are. And enzymes will cleave the DNA and pull one allele off one chromosome and move it to the other one and switch them. They'll switch genes. That's called synapses, crossing over. Synapses actually moves a gene from one arm to the other. I mean, and think about it. If dad's gene got a little, genetic disorder in it, what mutated, I would randomly move that mutation to mom's gene, right? So we're randomizing where all those mutations go back across all the different chromosomes once they're divided. So idea of crossing over is to increase randomization, not evolution, though. Remember that. That just increases the amount of variation. All evolution leads back to only one process. That's when the DNA divided. That's it. That's when you're putting the mistakes in. Everything from now on is about randomizing where those mistakes are at to generate the most variation possible and in the simplest way possible. Right? So tetrad formation, the arms touch each other. Now, is it a lot of genes? No. So I mean, a lot of times people think, oh, there's thousands of genes being moved back and forth. Not really. Not really. I mean, a few hundred get moved back and forth, out of the 23,000 that we have. Now, it's also not all of the genes that can get moved. Some of them don't. Now, I want you guys to think about this. Look at my arms where they are out here. So my arms. Now, look, I'm crossing over. See where my arms are touching out here, way out by my wrists? They're not touching down here, are they? They're mechanically restricted. They don't touch down here. And that's a good thing because what your body does, it puts all like the real critical genes here, like, this is my heart, my lung, my kidney stuff down here. You don't want those things randomly moving apart because you might make mistakes. Because it's a mechanical process of enzymes like moving DNA. It's like Amazon. How many times does your shit get lost every once in a while? You don't want that. So, if it's critical genes, they stay down low in the arms because you don't want them crossing over too much. It's too mission critical for you. But stuff that might be likely like arm link, leg link, amount of hair that you have or something, which, leads to variation, which is adaptable on the planet because of climate change and stuff going on. That stuff's on the outside up here, so it has a higher frequency of changing over. However, what a great system that is. I love that, right? Even the mechanics and the shape of the chromosomes are designed to enhance your fitness by allowing certain areas to cross over more and certain areas to cross over less, right? Okay, that's one level of variation that we get. We get a little bit of movement that way. But where is the action really at? Let's find out. Okay. Oh, by the way, so, once those chromosomes have crossed over and stuff, see, now you got a problem. Remember, we're double diploid. We don't just have 23 pairs, right? We have 46 pairs of 92 total chromosomes. Oh my God. That new cell is not going to last very long. And this is why this sort of process of cellular division here that goes to meiosis happens very quickly. It only takes a couple hours because all that cell or DNA is shut down. I mean, no processes are going on. It's like you holding your breath. You better do it fast before you die. So this is what's going on. Everything's organized and happened very quickly here. So the first thing that happens is that now we have too many chromosomes, so we have to immediately divide the cell. Okay? So the DNA lines up like in the center, right in the middle of the cell. Chromosome 1, 2, 3, 4, 5. We have double the amount, right? So we actually have two number, ones of each one mom and dad, two number twos of each mom and dad, two number threes. So the first thing that happens is that the cells divide and the chromosomes are pulled apart. And you can see the little lines that go to them. Those are special proteins, which we call telomeres. You don't need to know about that crap. It's mechanical. The chromosomes are pulled apart and they go into the receptive daughter cells, new cells, okay? All right, now here's the thing. You're only looking at one possible combination. Now you're looking at just two chromosomes there, right? See possibility one, it's the possibility one. We're showing two chromosomes. That's the chromosome 1 at the very top, mom and dad pair, and chromosome number 2, the one at the bottom. Then remember you have 23 different chromosomes, right? And here it shows, okay, all of dad's number 1 and all of dad's number 2 go into one cell. On the other side, it says, okay, all of mom's number 1 and all of mom's number 2 go in them cells. So you have little blue chromosomes and the possibility 1, a little red. How else can that happen, do you think? Thought about it? What happens if, in possibility 1, mom's red was not on the right side, it was on the left, okay? But the second possibility where dad's blue was on the left and mom's on the right. When the cell pulled apart, what you'd have is possibility number 2. See, possibility number 2. Where now you have daughter cells with mom's chromosome number 1, or dad's chromosome number 1, and mom's chromosome number 2. See, this is what we call Mendel's law of independent assortment. Those chromosomes can independently assort and go into different dividing cells. There's no which way it should be. It can one way or the other. And by doing that, we're getting different assortments of mom and dad's chromosomes. Remember, with different sorts of mutations that have happened to them already and different sorts of crossing over that have happened to them already. This is where variation is increased dramatically, okay? And that's what Darwin didn't know right there. He goes, I can see all this variation around it, but how the hell does it happen? You know, not only genetic change, but how do those traits get mixed up? Now, the thing is that when we look at the second round of division, you're down second rows, right? The chromosomes are still in pairs. Okay, we're back to a normal diploid cell. Oh, horrors. We've got to divide again. Now we have to divide one more time. Now, in the second round of mitotic division, we're pulling the chromosomes apart. Now we're just pulling the whole chromosome apart. We don't know which side of the chromatid. And each chromatid side has different mutations in them too, are going to be going into the gamete cells. So another reductive division, right, throws another higher level degree of randomization. So I'll tell you what the chances are, okay, of another organism like you, exactly copy like you, coming on planet Earth are, they're 10 to the 23rd. 1 times 10 to the 23rd. That's about how many atoms there are in the universe. That's the chances are. And it's probably smaller than that. We wanted to be safe. We wanted it to be safe, okay? So if you don't think you're absolutely unique, slap yourself. Say, Mercer said, just hit yourself a couple times. Wake yourself up, because you are absolutely and totally unique and special. Nothing like you will ever exist again, you know? So when people talk about the sanctity of human life, I look at it from a biological perspective. Like, hell yeah, you're like the rarest. Every time I see somebody go, you're the rarest thing that ever existed, because you are. Not that you're absolutely irreplaceable. I don't care what item in the world you find, what you think there's only one of, look at yourself in the mirror and go, I'm rarer than anything out there, right? So think about that. For anybody who wants to hurt themselves, stop your shit. I mean, just stop it now, because you got to realize that, right? And the variation which you received is absolutely critical for the survival of your species. And reality is that if you were not supposed to be here right now, you would not be. You are the end run of 4 billion years of complex evolutionary processes like this. So if you're not genetically supposed to be here, there's not a chance in hell that you would be here. And that's one of the purposes, if you've ever watched the film I want you guys to watch, Cube, right? You talk about people with Down syndrome, or people who are savant, or people who have different mental processes that you don't think are normal. You're absolutely wrong. Because remember, a group of those people would think you're abnormal too. Right? And we got to think about this too. What's the difference between a language and a dialect? A language is a dialect with an army and a navy. Okay? It just means you got more of them. It doesn't mean they're not as important. So all this variation that we see is critically important to the survival of the species, no matter who you are out there, no matter what condition you think you are. The process is a social comparison. And the one thing we know about social, right? Social facts dictate our reality, not the true scientific facts. And that's some of the most painful things that we're dealing with right now. And one of those side effects is racism, right? Which I can't even believe that the humans at this point are not a level of evolution, that there's dummies out there who actually believe that they're superior because of their skin color. It's just absolutely mind-numbing to me to see people like this. I just don't understand how they can actually exist. Or even, they probably end up shift leaders at Starbucks for the rest of their lives, right? Okay, so I'm going to go on. So this is the whole thing about meiosis. And the thing about it is Mendel predicted this. So you can never see this happening. And that's the importance of Mendel. Now people go, well, you know, Mendel didn't really discover that much. No, actually he didn't. He didn't. But what he did was he told scientists, okay, from the patterns that I see here, okay, they're going to be Q unit factors. And the cell, whatever you're looking at, whenever you go inside your body, you're going to find 2 things. And one of those things is going to be dominant to the other in this system. So when people started looking at the body in a microscope, you have no idea how big the body is in a microscope. Oh my God. I mean, it's huge. Like a cell would be like the Bay Area, you know? And the Golgi apparatus that like say packages stuff, we've got the size of your house. Think about how huge these universes are inside. And scientists need to have like homing beacons. What do things look like? If they did not have Q unit factors, both of them can be different. Scientists would not have known where to look for, and it would have slowed down our hunt for the genome. In fact, a guy by the name of Walter Sutton in 1902 utilizing one of the first high-powered microscopes, had just read Mendel's books, right? Mendel's works, his releases on it. And he knew to look for 2 unit factors. So when he was watching a cell going through, the division process, he noticed 2 things were coming together in pairs. And he goes, oh my God, that's it. That's what Mendel was talking about. So it allowed him to hone into what he was looking for. So that's the importance of Mendel, is to show you how science works, right? To show you completely, how science works. We build on the ideas of others. They give us ideas of where to look. And that's one of the problems I criticize many other science courses, is they don't tell you why Mendel's important. Now, Mendel has some shortcomings, and we'll talk a little about that here too. Now, Mendel's system, as we thought about, was a system of dominance and recessivity, right? That's his entire system. But here's the thing, that Mendel's traits, he was looking at his plant, were either or. So every trait he looked at, plant was either short or tall. There wasn't any in between. Plant either had, white flowers, or it had, yellow flowers. The peas were either yellow or they were green. The peas were either round or they were wrinkled. There was no in between. Because Mendel was mapping what we call discrete traits. And discrete traits are when we see one gene, one effect, okay? So one coding protein produces this one effect, okay? The shortcoming is that's not the way that you work. You don't have that many discrete traits. You just don't. There are a few. Like, anybody out there can wiggle their ears. I love that. I had one student wiggle their ears. It was ridiculous. Like, if you can do it to it, don't embarrass yourself. But once you're like wiggling it way up and down, I'm like, holy crap, how do you do that, right? Like, woo. I'm normal. See, right, what normal is. The thing about it is that if you have one gene that links, the one protein that causes the muscle to be linked to one little nerve ending here, you can wiggle your ear. But there aren't any halfways. There aren't any people who can do it halfway that you can or you can't. How about, this was disgusting, but like, I remember the first time I kissed a girl was a very long time ago, like 4 years ago. No, I was kidding, a long time ago. And she stuck her tongue out. She goes, I was disgusted. Like, I'm never going to kiss a girl again. It delayed my growth and development like 20 years. It was disgusting. Right? But and I realized there's only 2 different ways you can do it. People can go, they can't roll the tongue. Or people, there's no halfway. You can or you can't. Those are discrete traits, because you have that one gene that produces the neural connection to the muscle that allows it to do it. How about this? Let's try this. When you hold your hands like this, like you're in a ball like this with your thumbs crossed, thumbs like this. Either you can hold them one way or the other for a long period of time. Okay, you hold them out like below your lap. Cross your thumb across one way. One way will be comfortable. The other way, after about 10 or 15 seconds, it just starts bugging you like, I gotta let go, I can't do this, right? Well, that's because your bilateral symmetry, the bilateral nerves move in one direction or the other, and it's dictated by one gene. So that's a discrete trait. But you only have a few discrete traits, maybe like a dozen. The rest of the 23,000 traits are not Mendelian traits. They're what we call continuous traits. And this is where we have multiple genes create multiple effects. Okay, we'll call those polygenic traits. So in the real world of evolution, when we do a mutation in one spot, we are affecting a network of genes that are working together to create a trait. And they create a continuous sort of distribution of characteristics. For example, how about height? When have you seen humans come exactly in 3.5, 4 foot, 4.5, and 5 foot models? There's no like real stuff. We are all continuous. If I lined you guys up on the classroom, you'd see all continuous distribution of height. That's because dozens of genes come together to actually create something like height. Or even eye color. People go, well, what color eyes you have? We have blue, black, brown, green, like, wait till you look. If you really look in someone's eyes, you'll find a continuous distribution. There's no just one blue. It continues all the way into greens, and switches to bright browns and dark browns. That continuous variation is caused by dozens of genes. Thus, when we are mutating these genes, we're getting a rich tapestry of effects because they're polygenic. They come together to create far more complex blends. So if the world operated on discrete traits like Mendel, even though this process of meiosis is happening, we would not see the incredible amount of diversity we have in the world. But because in reality, this system is working on polygenetic effects where many genes are working together, we see far more variation than we could with simple Mendelian traits. So when we talk about Mendelian traits, I want you guys to think about simple discrete traits. He just started to undermine the very simplest rules of which we've taken off far from it. Right? But he pointed the direction for us to be able to look. That's the importance of it. Okay. All right. Okay, so last thing I want to think about too is in reproduction. Because we're animals, it doesn't work this way, you know, in all the species of life which is out there, but for the majority of all 4,000 mammals, it sure does, and even reptiles too, is we have to think about what happens to the gametes once we actually produced. So once we're through with meiosis, maybe we have 2 rounds of division, you know, which create more variation through Mendel's law of independent assortment because the chromosomes can go in all sorts of different combinations together. You end up with 4 daughter cells, 4 haploid daughter cells. Right? Well, the thing is that it happens essentially the same way in males and females to a point. Once we have the 4 daughter cells as they begin to be created, in females is very different. What happens is because, you know, the primary gamete, which is in the female's body, has to stay there for a very long time. It's there by the time you're 13 months old, and it has to be there in good health, you know, until you go into reproduction, which, you know, hopefully if you're a female, try by the time you're 36, because that's the line in which that gamete is going to begin to fall apart, and you're going to have serious problems with probably some birth defects, right? 36 is the touch-off line. Don't get sold on this 40-year-old women, they're just fine. Don't run that risk, okay? What happens is because it has to last for so long, one primary egg is chosen. And I may chosen is all the other eggs, all the good stuff is moved over to what we call the primary oocyte. Okay, that's what happens is all the good egg is chosen and reinforced. And then what happens is the other eggs, because they become depleted, the DNA is shut down. They become what we call polar bodies. They're actually proteins that bind the DNA so they can't go into reproduction. So only one gamete of the female survives, is the primary chosen egg, right? And that's the one that's loaded, of course, you know, in the fallopian tubes. It sits there and is supposed to be nourished and is healthy. Okay? How about males? It's different because this is a continuing, ongoing process where the gametes are constantly being made. None have to be stored. In fact, gamete turnover is millions of times per second in the testes. If you even take a hot shower, you're killing millions and millions. If you're going to a hot tub, you're killing millions and millions. Don't worry, you can't sterilize yourself because you're going to produce more if you're a male, continually. Now, the thing about it is that that DNA that's being packaged into the sperms, you know, has to go to work. But think about the type of DNA it is. It's different. We got to think about this. The DNA that's packaged into the sperm for males is nothing but nuclear DNA. Nuclear DNA, that's your nuclear DNA. But isn't there DNA in 2 parts of your cell? Yeah, remember I said that the power stations of your cell were known as mitochondria, right? And the mitochondria are living symbiotically. You know, they're the downlines of billions of years of evolution, but at one time, there were 2 domains of life. One was called archaeans and one were called bacterium. They were 2 single cell levels of life, simple. And it was pretty boring until one day the archaean ate the bacterium. And there were genetic changes to both of them. And the bacterium began to survive inside of the archaean and reproduce together. And the bacterium became the energy producing member of the cell, our cell, and created a new type of cell called the eukaryotic cell. So since that time has happened, that bacterium has changed. It's become the mitochondria of your cell. But because it was a free living organism, it has free living, freestanding DNA. So you have DNA in 2 parts of your cell: the nucleus, nuclear DNA, and the mitochondrial DNA. So when male sperm are being made, only the nuclear DNA gets packaged into the sperm. That's it. All of the mitochondrial DNA, on the other hand, is over in the female. Because when that primary oocyte is being made, it gets stuffed with mitochondria. All the mitochondrial DNA that I have in my body in any male alive today comes from their mother. Because the male has to fertilize that that gamete where the mitochondrial DNA is already at. So we can actually track human migration and evolution just by looking at changes in the mitochondrial DNA. But what we're doing is we're tracking women. We call that mitochondrial Eve, by the way. Have you heard that term? So we're always looking for the genesis of the human race by trying to find the earliest members, which we have located in Africa, which have the least amount of changes to that mitochondrial DNA through the female line, right? But that's an important part to remember, that all your mitochondrial DNA comes from your female side. If you're female, you're harboring that DNA too, and not the males. But in the same way as nuclear DNA has to replicate itself, well, so does the bacterial DNA too, as it goes into, not the bacterial, but the mitochondrial DNA has to replicate itself too, moving from, primary sex cell into, primary oocyte or the gamete. So all the genetic changes that are happening, within our species and the mitochondrial DNA is happening within the females. All right, okay. So, here. This is my warning slide. Don't let it happen. That's cost $250,000 right there, right? Don't let it happen. Don't let it happen. Right? It's fine. If you don't want to spend the money, children are fine. But remember, now what we had before were 2 haploid cells. Now, what do we have? Somebody speak it out. What kind of cell do we have now?
A diploid.
Yes. Awesome. All right. So that's as much as I wanted to talk about today, right? I know once I go past an hour on this stuff, it starts to get, we hit just about the hour point. So I'm going to stop here for any questions you're thinking about.
Was that the end of the slideshow?
Yeah, that was in the slideshow. Yeah.
Okay, cool. Thank you.
Yeah, I want to keep that short and just get to the basic principles, because generally what happens is while I love your lab manual, she wrote this as her PhD thesis at Cal. Okay, this is when she was at Berkeley. So remember, this is like for a Berkeley laboratory group, which is why there's 16 labs in here. I'm only giving you guys a small selection of them because other ones are really hard. And even this one has more information you probably need to know. So when I give you my slideshow, I'm sure you're showing what you really need to know for the quizzes. All right.
All right. Will you go over the second lab like you did for the first one?
Yes, I am. And so what I'm going to do, and what I decided to do, I think is easier, is this lecture is being recorded now. And right after I finish this lecture, what I'm going to do is copy the lab out, and then I'm going to do a PowerPoint walkthrough. So I'll actually show you how I actually lined everything. I'll do a little writing board, so you'll see that. And it should be up in about an hour or so. How's that work?
Okay, that sounds good. Thank you.
Yeah, I thought it worked easier for you. And then next week, if you have any questions about that, we can talk about it.
All right, sounds good.
Cool. All right, you guys. I want you guys to have a wonderful afternoon. I'll talk to you guys later.
Thank you. Have a good day.
Thank you. Goodbye.
Bye.